Alright, yeah, hey, I'm like creating this video for for people who kind of want to get a very simple introduction to like protein structure and proteins. If for why would you be interested? Yeah, for like, I mean, kind of more people who are coming from the machine learning side, and or mathematical side, or data analytical side, who want to understand this just so that like they know enough to kind of dive deeper. I will link resources below in the video, and also kind of if they want to just work on like one of the frontier models, that this kind of enables them to have some understanding so they can do this. Yeah, so I structured the video in three, in kind of like three major topics. The first one will kind of just give you the basic understanding of, or like will give kind of the very basics of proteins. The second will be like how or why is this even possible computationally, and then I will kind of go through, I guess, like the where I guess the interest in, or like the idea that we can even predict this computationally, and then lastly, kind of third will be categorizing proteins. So there are substructures, in kind of like linearizing or separating the more complex problem into more solvable sub problems. Yeah, let's get started. So I think, and I'll show the pictures here. So amino acids, you kind of go from DNA to RNA to then the protein. On the RNA, there's like some protein machinery, or like some machinery. Every protein is kind of like proteins are the major function, providing the major functionality in biology, and you have RNA, for example, also, which provides like, to some degree, some value, or like, some function, but we're not talking about RNA in this video. This will be another one, and yeah, so then this gets translated. You have like all of them are connected by some so you have the RNA. The RNA is like, it has a start and it has an end, and this has signals, and then there are codons. Codons consist of like three RNA nucleotides, and those codons, they encode like some the final thing, which will be in the protein, which is an amino acid. You can just imagine it as like a Lego, and this is kind of then connected with another, and then another, and like this is a code, like so it goes from one side to the other, like a machinery which translates the codons to the proteins, and this like forms a chain, and this chain then like folds into some 3D structure, and in some 3D structure, there are different types of the amino acids, and each of them has like a specific characteristic, as you can see here. And then relevant, and then you kind of there's like this model, which is you can categorize it into primary, secondary, tertiary, and quaternary structures, and this is kind of just a model, because while it translates, it also automatically folds to some degree. And yeah, but the primary is just means you look at the plane, the sequence of the amino acids. And this is really just like an alphabet, and you also look at sometimes at the like sulfate bonds, which also happened. So this is the first part, kind of like the rough overview of the proteins, and then yeah, so then computationally, why is this relevant? Because the structure contains the main functions, and it has different sites, which is like the active site, for example, and this is where for example, in metabolism, actually the actual function happens, and the large architecture is not where the function happens, but this is not that relevant. And a very good finding was that you have this, like it was Anfinsen's dogma, that all information for the folding is contained in the sequence itself, which is a surprising finding, but it's like very old. And then there's also like post-translational modification, which I won't be going into, but those are also, of course, very relevant. And the other finding was the folding happens systematically, and there's one energy state which is the lowest, and that's where kind of the functionally folded protein is in, and kind of the Leventhal paradox, if you want to call it a paradox, then it kind of has the answers the issue that what they are saying is basically that the protein folds, and there's it folds in a systematic way. So I mean, if you have this long chain, and for each chain you have like different torsion angles, one is phi and one is psi, and then those can rotate freely, and even if you would say you constrain this to like, let's say, 3 states. For proteins are relatively long, so you have a chain of like 200, and you have so many possibilities that you just cannot compute it. There's also the omega angle, which is from the carbon to the oxygen, but this is usually just like 180 degrees. And the other thing, so you have all of this, and then kind of you have the classification of amino acids, which I mentioned earlier, the Lego blocks, and that they have different characteristics, and most of them are just kind of for the architecture, so they are like mainly similar ones are used, but then there are also very few where where their like function is determining the output, and like determining the function of the protein. And so this architecture, it works mainly, and so that as like Ramachandran plot, which is kind of you have on the x-axis, x and y axis, the phi and psi angle, and then you can kind of see the cluster together. I'll show a picture here, and a cluster together, and there you can see that and kind of he studied then this, like, why is there such a cluster, what does this mean, and then he figured out this is like the alpha. He kind of categorized it mainly the alpha and beta sheet. Both of them have like 1/4 of them. So in all proteins, the amino acids, all amino acids in proteins, like 33%, or like roughly one third of them are in alpha sheets and 1/3 of them are in beta sheets. Yeah, and then there's like other small categories as well. There's loops, which is kind of, you have one and then it turns around. Then there's also like coil, which kind of don't fall into the other categories. That's, I guess, how it's classified. You also have this like super secondary structures, which is kind of, you have a coiled coil. Like coil was how alpha sheets used to be named. So then you have them coiled together, like 3 alpha sheets, which like kind of twist into each other, and also like the beta sheets can form like a coil as well, by kind of clustering together. I showed the 2 pictures here. You can kind of class the protein structures as well. There are a lot of methods to do this, and one of the very popular ones is the CATH, which is for architecture, the T for topology, so kind of the way the string goes. The architecture just means how it generally looks, and the H stands for like the homology, and then there's like 2 additional things, which I will also link below. Like one is mainly the C, I guess, stands for composition. Like a main is the one which is mostly alpha sheets, one which is mostly beta sheets, and then there's like a mixture of both of them, and then there's the ones which have like other structures, and then it kind of fans out, and there's a lot more subcategories. Also, what you need to know is that this kind of introduces like there are things which are then wrongly classified, and so people have tried like to use variational autoencoders to improve this, and then once you have a secondary structure, so then we kind of go to the algorithms, kind of the baseline algorithms. One of the most important to have this secondary structure is the DSSP, which kind of uses the energy of like hydrogen bonds, and kind of says if it's like lower than a certain value, then you put it in one category.